Hello everyone, welcome to my YouTube channel. Learn with me, Dr. Ram. Simple Learning Part 1. Yes, today topic is complement system, which will confuse easily. In complement system, there was three pathways: classical pathway, Liston-Manos pathway, and alternative pathway. First, we will see the classical pathway, where there is antigen-antibody interaction, whether IgG or IgM mediated antigen-antibody interaction happens, and C1 activation will be done, and then C4 activation. When C4 is divided into C4A and C4B. C4A went into the opsonization, that means degrading, and C4B again act upon C2, and C2 is divided into C2A and C2B. The only place where C2B went to the opsonization is C2. C4A go to the opsonization. Then again C2A act upon C3 and go for divide into C3A and C3B again. Now C3A go for opsonization and C3B again go into divide for C5. Act upon C5 and C5 is divided into C5A and C5B. C5B is act upon C5, C6, C7, C8 and C9. At the end, it will enter into the membrane attack complex, breakdown of everything or breakdown of the antigen. Now, this is known as classical pathway. First, if you understand this, then we can go into the Manos binding pathway. In Manos binding pathway, there is also called as the lecithin pathway. Manos binding complex is activated first, and from there C4 is activated initially, and again C4A and C4B is going to divide, and C4A go for opsonization. C4B now act on the C2, again the same cycle continues. So this is known as lecithin pathway until C2. There is no C1 C4 directly acting on C4. Manos binding C4 and C2 same. And in alternate pathway, this is slightly different, where C3B initially there is no C1 C2 and everything. First it starts with C3B and act upon the factor B and divide C3 small b and capital B. Again act upon factor B and it will divide C3 small b capital B small a. C3b small b capital B small b. Just simple friends. Here C3b is divided into A and B, and here it is A go for opsonization and B is there, and again B divided into BA and BB. Now this B capital B and small b under the action of properdin. This is the main important enzyme. Properdin, it will go into the again act upon C3 and the same cycle continues. Alternative pathway, before some pathway is undergoing and continue from C3. There is no C1 and C2 and C4 for alternative pathway. And classical pathway, antigen-antibody interaction. Manos binding pathway or lecithin pathway, Manos binding complex is the initiator, C4 and C2 same. And out of all this, what are the powerful opsonants for the opsonization is C3A and C5A. Okay friends, thank you. Hope you like the content. Please like, share, subscribe and comment. Thank you friends.